Alors bonsoir à tous. Bienvenue à ce forum ce soir, un forum qui va être extraordinairement intéressant. Good evening, everyone. We have an extraordinary panel today. We are blessed, privileged to have those three individuals, fantastic experts on the stage to engage, to create a conversation around gene editing. That conversation is important because gene editing will have an impact for many of us. It will change the course of some medical therapies. It will change the course of some treatments. It may, in some cases, improve our quality of life. So this topic matters. That topic is important. I'm grateful that we have this conversation, like you said, Monica, early on in the evolution of the technology, such that we don't repeat, hopefully, the story that we have seen unfolding over the last 25 years for genetically modified food. I was involved myself in some of the science of genetic modification in food crops, and I was involved in the debate, and certainly the science community early on was frustrated that they were not able to engage with decision makers, engage with the public in a conversation about the possible impacts of that technology, those technologies. And once the genetically modified food debate took off, again, I think we had a sense of frustration that the science community was unable to engage with some of the players. And some regret, I think, that some of the decisions that were made were not quite as evidence-based as some of us would have liked. So it's absolutely fabulous that we can have this conversation tonight. So thank you for your interest. Merci d'être ici ce soir. C'est une indication de votre intérêt dans un débat, dans une conversation informée. The format tonight is awfully simple. Each panelist will talk for about 10 minutes, and then it is a conversation with you. It is a conversation fueled by your questions. I have to admit that no matter whether I was moderator or not, I would have been here tonight because the conversation is interesting. Now, being a moderator, I suppose I get to ask all my questions and I'm just kidding, rather. The 10 minutes each for each of the panelists is to set the stage for that conversation. So first, we have Dr. Janet Rossant. Again, like Monica indicated, I will not read the bios. You have them with you in your chairs. She's senior scientist, developmental and STEM biology at SickKids, University of Toronto, as well as president and scientific director of the Garner Foundation, and recipient of the L'Oréal UNESCO Prize for Women in Science. So congratulations. Setting the stage then for Dr. Jennifer Kuzma, who's Fulbright, visiting research chair in Science and Society here at the ISSP for a few months. Co-director of the Genetic Engineering and Society Center at North Carolina State University. And Dr. Kuzma will build on the introduction that Janet will have provided to talk about policy issues related to gene editing, the risk associated with those technologies. And finally, Dr. Rob Annan, Vice President, Public Affairs, Genome Canada, and formerly from MITAPS, will provide the context in which policy and regulations are developed in government. How do governments wrestle with, I would say, complex problems? So now I will turn to Dr. Rossant for an introduction on gene editing.
Thank you. So I think we just heard from Mona a very good summary of everything that we're going to be talking about tonight. And she also said that gene editing and genetic engineering is not new. I would go further. I would say that if we think about genetically modifying animals and plants, Humans have been doing this ever since the dawn of agriculture, ever since we domesticated animals. Just think of some of the examples. Think about the difference, those of you who know about plants, between teosinte, which is the wild plant that gave rise to corn on the cob today. Teosinte has little tassels. Corn on the cob is a great big thing that we all love to eat. Or think about what we've done to cows. Look at the Holstein cow. It's a milk machine. And we've done that by genetically engineering. But we don't do it by gene editing. We've done it over the centuries by just genetic selection. We look for those animals or plants that have traits that are beneficial. We select those, we interbreed them, and over time we see changes that have really been instrumental to humans being able to essentially become not just hunters and gatherers, but agricultural human beings that we see today. And of course, don't even think about dogs and all the genetic variation in dogs. So this is gene selection. So why do we not think about that as being gene editing? Well, because somehow this is a natural process. And it's certainly true that with the tools of being able to actually directly modify the DNA, we are moving from looking for natural selection to do the job for us to being able to understand more about genes and how to direct genetic changes much faster than we could do by the old selective process. We can speed up and directly genetically modify animals, plants, microbes, and yes, potentially humans with the new tools of gene editing. The power of gene editing really has unprecedented potential for genetic enhancements. But as we've heard already, that power, the speed and the efficiency that we can now do these genetic changes brings about concerns about risks and limitations that we need to put in place to protect ourselves from unpredicted consequences or indeed directed misuse. So what is gene editing? Well, again, we've been editing in experimental situations quite a lot over the last 20 or 30 years. We can make alterations in the genes of cells in culture. We can do it in mouse embryos. I've been doing that for a long time in my lab. But what has changed over the last few years is the new tools of gene editing, particularly what's called CRISPR-Cas9 gene editing. You may have seen and heard about this. This is a system that is not very different from other things we've done and used over the past to edit the sequence of our DNA, but it is a game changer because it is incredibly precise and incredibly efficient, so much more than any of its predecessors. So what is it? Well, there's a nice serendipity story about how CRISPR-Cas9 became the tool that we all use today, and it has to do with yogurt. So yogurt is really the basis for why we have CRISPR-Cas9 gene editing, because when you make yogurt, it's very important that the yogurt bacteria is very healthy, otherwise you don't get good yogurt. So Danone, the yogurt company, employed a large number of microbiologists and actually had partnerships with microbiologists around the world, including here in Canada, particularly Sylvain Moineau at the University of Laval, who were working on trying to keep bacteria healthy. Now, bacteria get infected by viruses just the same as we do, but the bacteria has developed an immunity system to get rid of those viruses. And that bacterial immunity system is based on recognizing a piece of DNA that comes in a virus, lining up a piece of RNA against it, and then bringing in an enzyme, a nuclease, that will cut the virus right where the guide RNA hits and kill the virus. So it's a way of the bacteria getting rid of viruses. And of course, what you want is healthy bacteria, so you really need to make sure that the bacteria has a functioning immune system. Now, what happened is that process, that immune system, that nuclease and the little guide RNA that really makes the precise cut, then was taken by a number of scientists, biochemists, who did indeed receive the Gairdner Award, as did the bacterial immunity guys as well. The biochemists took this and turned it into a tool that you could edit not just a bacteria or a bacterial virus, but you could use it as a toolkit to edit any cell you like. And the toolkit simply works by taking a piece of RNA, a guide RNA that is complementary to a piece of DNA you want to target in your cell, bringing the nuclease, it cuts, and after you've made that precise cut, you can add in other genes. You can repair genes, you can knock out genes, you can insert things in genes, and again, it's incredibly precise and it's incredibly efficient. So it's that tool that has really changed a lot of the ways many of us do research today. It's an incredibly important research tool. People are using it to screen for genes that are involved in cancer. People are looking at immune systems. I use it to study developmental biology. But it also has enormously important potential applications that are going to affect us as humans going forward. So I'll just give you a few, and we can also talk about more as we go forward. In agriculture, you can use gene editing, CRISPR-Cas9 editing, to think about making improved disease and resistant crops and in disease-resistant animals. In biotech, there's a lot of interest in gene editing to make microbes that can make new chemicals. So we put new enzymes and new functions in microbes. Or we can make better microbes that can do bioremediation and clean up some of the messes we as humans make. We can turn CRISPR back against the microbes itself. This is being done by one of the founders of the bacterial immune system. So you take the CRISPR system and use it to develop new antimicrobials to hit antibiotic-resistant microbes, using the whole system again. More controversially, you may have heard about gene drives. This is an additional process whereby you can use gene editing and make it such that it drives a genetic change through a whole population very fast. I can explain it, but it's a bit complicated. Suffice it to say that people are looking at this as a way, perhaps, of getting rid of malarial-carrying mosquitoes. You could put in a gene, makes the mosquitoes sterile, and have it rapidly pass through the population. But there are some real concerns about gene drive. How do you control this if it's spreading so fast? How do you know that you won't have unintended consequences? How do you control population spread, etc.? So this is a very controversial area. So these are just some of the broad applications we can think about. But in direct human health applications, we're seeing things moving towards the clinic in a number of areas today. Thinking about really repairing genetic diseases where we know what the genetic cause is. Can we go back and repair the gene? So diseases like sickle cell anemia, beta thalassemia, we can take potentially the blood cells, the stem cells from patients with these diseases and repair the mutation and do it so efficiently that when we put those cells back, they can produce enough normal blood globin, hemoglobin, to essentially help those patients do much better and in fact perhaps cure them. We're all even thinking about taking diseases like Duchenne muscular dystrophy, which is a muscle-borne disease, and taking viruses to deliver the gene editing tools right into the muscles of the patients. Proof of principle in the mouse, this works. I know we've cured mice a few times, I do understand that. But in this case, I think there is real hope that this may have clinical applications in a not too distant future. Preventing HIV infection. Modifying the receptor for HIV on the T cells of patients who are infected with the HIV virus. This is going to the clinic now. Generating T cells, modifying killer cells, killer immune cells to kill the patient's own cancer by CRISPR-Cas9 editing. So these are enormously important and exciting applications in the clinic. It's moving very fast. There are many modifications of CRISPR-Cas9 as a research tool and as potentially a tool for applications in things we can't even think about. It's pretty clear that the public understanding or the regulatory environment in which we're working is really having a hard time keeping up. Human gene editing in particular has gained a lot of attention because it has raised this issue of designer babies that Mona mentioned. The concept that if we can really do gene editing so efficiently for genetic diseases, why would we let people be born with genetic diseases? Why don't we go back into the embryo and fix the gene before the embryo grows up to be a human being. So prevent genetic disease rather than trying to treat it after the fact. So this is called germline gene editing, because if you do that alteration is going to be inherited in the next generations. Now, in my own research in the mouse, we've actually just recently developed a new sort of modification of the CRISPR approach, where in the mouse embryo, we can actually add genes, alter genes in the embryo itself at an efficiency that approaches 100%. We never used to be able to do that. Maybe 1% of the embryos would be modified, in which case you wouldn't want to think about doing it in humans. We're close to 100% in the mouse. Then we have to start at least discussing whether we should be thinking about doing this in humans. But what would we do? How should we consider this? Where do we draw the line between editing to prevent serious genetic disease and genetic enhancement? Healthy babies versus designer babies. These are the kind of questions I think we're going to be discussing today. I'm certainly not going to give you any answers. Suffice it to say that I think as scientists, it's our duty to help explain the science, but also to get involved in the ethical and regulatory debates, because these are serious concerns. We do need society to think about how we see these technologies being used in the future, how we develop a legal and ethical framework that will allow beneficial uses of gene editing while balancing individual, population, and even ecosystem risks. So I'm looking forward to our discussion ahead. Thank you.